Hello everyone. Today we will discuss about de novo synthesis of fatty acids. De novo synthesis means new synthesis of fatty acids. The major fatty acids formed is 16 carbon saturated fatty acid, that is palmitic acid. Fatty acid synthesis occurs mainly in liver and lactating mammary glands and also in adipose tissue, brain and kidney. Fatty acid synthesis occurs in cytosol and the major substrate for fatty acid synthesis is acetyl-CoA. In conditions like high blood glucose level, high citrate, high ATP and in well-fed condition, the synthesis of fatty acid is favored. Fatty acid synthesis occurs in three phases. First phase is transport of acetyl-CoA from mitochondria to cytosol. Second phase is carboxylation of acetyl-CoA to malonyl-CoA. And the third phase is reactions of fatty acid synthase complex. First phase is transport of acetyl-CoA from mitochondria to cytosol. As it is impermeable, acetyl-CoA cannot cross the mitochondrial membrane. So it combines with oxaloacetate by the enzyme citrate synthase and it forms citrate in the mitochondria. This citrate can cross the mitochondrial membrane. So it reaches cytosol. And in the cytosol, it is broken down to acetyl-CoA and oxaloacetate by the enzyme citrate lyase. This acetyl-CoA serves as the substrate for fatty acid synthesis. Then, by the action of enzyme malate dehydrogenase, oxaloacetate is converted to malate. Then malate is converted to form pyruvate by the enzyme malic enzyme. Here, NADPH is generated along with carbon dioxide. Then this malate can cross mitochondrial membrane and it reaches mitochondria and then convert to oxaloacetate by the enzyme malate dehydrogenase. Pyruvate can also cross the membrane and in mitochondria pyruvate is converted to malate. So in this way acetyl-CoA, which cannot cross the mitochondrial membrane, reaches cytosol and utilized for fatty acid synthesis. The second phase of fatty acid synthesis is carboxylation of acetyl-CoA to malonyl-CoA. Here, acetyl-CoA combines with carbon dioxide in presence of enzyme acetyl-CoA carboxylase and biotin and forms malonyl-CoA. Here in this process, 1 ATP is utilized. This enzyme acetyl-CoA carboxylase is the rate-limiting enzyme of fatty acid synthesis and the conversion of acetyl-CoA to malonyl-CoA is the committed step of fatty acid synthesis. Phase 3 is reactions of fatty acid synthase complex. Fatty acid synthase complex is a multi-enzyme complex and it is a dimer which is composed of two identical subunits. Each subunit has 7 enzymes and 1 acyl carrier protein. The functional division of fatty acid synthase complex has 2 subunits. Domain 1 of functional subunit is known as condensing unit and it consists of 3 enzymes: ketoacyl synthase, acetyl transacylase and malonyl transacylase. Domain 2 is reduction unit. It has also 3 enzymes and 1 acyl carrier protein. 3 enzymes are dehydratase, enoyl reductase and ketoacyl reductase. Then domain 3, which is the releasing unit, it consists of enzyme thioesterase. So the reactions involved in fatty acid synthase complex are: first one, condensation reaction, which involves domain 1, that is the condensing unit, step 1, 2 and 3. Step 1 involves the enzyme acetyl transacylase, step 2 involves malonyl transacylase and step 3 ketoacyl synthase. Step 4 is reduction reaction and it involves the enzyme ketoacyl reductase. Step 5 is dehydration and the enzyme is dehydratase. Step 6 also reduction. Here the enzyme involved is enoyl reductase. So there will be repetition of steps 2 to 6. That is 6 times. Then step 7 is releasing step. Here palmitic acid is released by the action of enzyme thioesterase. So let's see the steps of fatty acid synthesis in detail. Step 1 is addition of acetyl group to acyl carrier protein by the enzyme acetyl transacylase. Here this is a fatty acid synthase which has ketoacyl synthase of first monomer and acyl carrier protein of second monomer. Here acetate group of acetyl-CoA molecule is transferred to the SH group of acyl carrier protein by the enzyme acetyl transacylase and form a temporary molecule. From here, acetyl group is transferred to the SH group of ketoacyl synthase and there will be formation of acetyl enzyme. So that is step 1. In step 2, there will be addition of malonyl group from malonyl-CoA to SH group of acyl carrier protein by the enzyme malonyl transacylase and it forms acetyl malonyl enzyme. In step 3, there is condensation of acetyl and malonyl group by the enzyme ketoacyl synthase. There will be release of carbon dioxide and form acetoacetyl ACP which has 4 carbon atoms. Step 4 is reduction by the enzyme ketoacyl reductase. Here NADPH is required and forms beta hydroxyacyl ACP and NADP. Step 5 is dehydration by the enzyme dehydratase. There is removal of one molecule of water and forms enoyl ACP. Step 6 is second reduction by enzyme enoyl reductase. Here NADPH plus H plus is required and forms butyryl ACP and NADP. Then there will be a transfer of carbon chain from ACP to ketoacyl synthase and forms acyl enzyme. So this site is now free to accept the malonyl group of malonyl-CoA. So after 6 reactions of step 2 to 6 in fatty acid synthase complex, in first cycle 1 acetyl-CoA, 1 malonyl-CoA, 2 NADPH and 1 ATP are required. And for 6 cycles from 2 to 6, 6 malonyl-CoA, 12 NADPH and 6 ATP are required. So in total, the requirements of 1 palmitic acid synthesis are 1 acetyl-CoA, 7 malonyl-CoA, 14 NADPH and 7 ATP. Coming to step 7, it is release of palmitate by the enzyme thioesterase. There will be formation of palmitate. So the products are 1 palmitate, 14 NADP, 6 H2O, 8 CoA and 7 carbon dioxide. Further elongation of fatty acid chains occur in the smooth endoplasmic reticulum by the enzyme fatty acid elongase. Here, by the addition of 2 carbon units, palmitic acid forms stearic acid which is 18 carbon. Here 2 carbon units are provided by malonyl-CoA and NADPH supplies the electrons. Desaturation of fatty acids occur in smooth endoplasmic reticulum by the enzyme desaturases. Here NADH, molecular oxygen, enzymes like cytochrome B5 reductase, desaturase and cytochrome B5 are required for the desaturation. By desaturation, palmitic acid forms palmitoleic acid. From stearic acid, oleic acid is formed. So this is about de novo synthesis of fatty acids. This is today's topic. Thank you for watching.